Everyone, welcome. So today we will be going ahead with the chapter Biotechnology Principles and Processes. I will be dividing the chapters into 8 parts while teaching. One is introduction. Next will be tools for RDNA technology, where I will be speaking of restriction enzyme. Then we will be talking about cloning vectors and multiple cloning sites. Then after that, we will be talking about transformation of RDNA into the host. Then we will be talking about process of RDNA technology, bioreactors, and finally downstream processing. So now the first chapter we are going ahead. Let us get introduced. What is biotechnology? Bio means the use of biological or the life process. Bio is basically life. Technology means the how we are going to solve the problems, and all those we are going to talk about. How do we produce a useful product? Now, to state about biotechnology, it's a science that uses living organisms or the products of living organisms to benefit humans and their surroundings. Biotechnology it has got various application, be it in the medical diagnosis, be it in the environmental and energy management, plant agriculture for the improvement of crops like BT cotton or hybrid varieties, for RDNA technology that will be going ahead, for fermentation technology. Also, we use biotechnology, food technology, improvement of food technology, preservations, and all those things, health and pharmaceuticals, vaccines, then animal breeding and agriculture. Also, we go ahead with biotechnology. So, biotechnology, it basically deals with microbe mediated processes, making of curd, bread, wine, etc. Then we also deal with in vitro fertilization, where we talk about test tube baby programs and all those, the synthesis and using a gene for various activities, or we if you want to introduce a specific gene to get some desired characters. Then preparation of DNA vaccines, we use it, and correcting a defective gene also we can use it. So, for definition of biotechnology, European Federation of Biotechnology (EFB) has given the definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. So, the definition is given as the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. So whenever I'm talking about biotechnology, it is an amalgamation of biology, chemistry, and engineering. If we talk about biology and chemistry, it's biochemistry, chemistry and engineering, it's chemical engineering. Biology and engineering would be bioengineering, but when all of them is put together, I talk about biotechnology. The two core techniques, or I can say the principles of biotechnology, is genetic engineering and bioprocess engineering. In genetic engineering, we deal with genetic manipulation, modifying the genetic material for the desired trait. Whereas bioprocess engineering, I'm talking about the advancement of pharmaceutical industry, like production of antibiotics, enzymes, vaccines. So it is the final product that I'm talking about when I talk about bioprocess engineering. The steps in genetically modified organisms, which would include identification of DNA with desirable characteristics, introduction of identified DNA into the host, and maintenance of introduced DNA into the host, and transfer of the DNA progeny. So I'm just in details I'll be discussing in the next video. Hence, just to give you how this RDNA technology came into existence was because of Stanley Cohen and Herbert Boyer. They isolated an antibiotic resistance gene from a plasmid of Salmonella typhimurium, which was linked with plasmid vector and transferred into E. coli. As a result, the gene was expressed and multiplied in E. coli.